It's really a great honor and pleasure to have been asked to chair this session of this extraordinary 50th anniversary celebration of the PDB. Maybe just before I start, I have a few housekeeping matters to tell. After each speaker, there will be a few minutes for questions and answers. Please post your questions and answers in the Q&A just to the right of the talks. If you have any technical problems, just please click on the chat and you can ask an ASBM person for help. And this session, just let everyone know, is being recorded and it will be available until the 21st of May. So maybe I just could mention that as Helen said in her beautiful, beautiful opening talk yesterday, I was also closely involved in helping to establish the PDB in 1971, and then following Walter Hamilton and Tom Ketzel's footsteps 2 decades later, served as its head, as Celia mentioned. I just want to give a tiny view of how I see the meeting, and there have been a number of questions. What is the future directions of the PDB? So bear with me for about 2 minutes. I was lucky to have been able to do my PhD in Cyrus Leventhal's lab, who was one of the real pioneers in protein folding. And he thought out of the box a lot. I was also fortunate then to be able to work with some extraordinary colleagues, Helen, Ned Seaman, and Sung-Ho Kim, where we determined the structure of the largest RNA molecule determined at that time. It was a dinucleotide monophosphate, UPA. I mentioned this as we were also able to use some of Cy's very early but very powerful molecular graphics, and we made a morph of this crystal structure of UPA, which had 2 molecules in the asymmet unit, because we wanted to show that they were quite different conformations, and the morph showed the change that was possible for RNA structures. And I was able to show this at an ACA meeting that summer of 1971. This movie, another one that I made with Shoshana Vodak, who will be chairing the next session, and that was on crystallography can even be fun, are both available on YouTube, and you can see the links in my biography. I think the reason I bring all this up is that I feel that molecular conformational dynamics has been a theme throughout this symposium. I think it's in some ways, it's the major direction which the field and the PDB is going. It's clearly not enough just to see a singular high resolution 3D structure of a molecule. But as we've seen in the giant molecular complexes that Professor Rao and Professor Wu showed yesterday, it's crucial to be able to see how these change in shape in order to try to understand their function. And in parallel, there's a whole class of molecules that are just absent from the PDB. These are the intrinsically disordered proteins, because it's hard to see a structure of them, except sometimes when they complex with other proteins. My feeling is these molecules in the future are going to play a great role, and maybe in some of the tools that Professor Bonvin mentioned yesterday will be able to help elucidate what their roles are going to be and how their structures form in complexes. So enough said about the background, but I think that's the future of to see, as Dino Moras once said, to see molecules dancing in the cell.